Question of molecular basis of inheritance. A part, identify the process. This process is prokaryotic transcription, and the enzyme, name the enzyme catalyze this process is RNA polymerase. And what are the additional complexities in eukaryotes for this process? In eukaryotes, hnRNA undergo additional processing called as splicing, capping, and tailing. In splicing, the introns are removed and exons are joined together. In capping, methyl guanosine triphosphate is added to the fifth end of hnRNA. In tailing, it's added to the third end, third prime end in the template. The fully processed hnRNA is called as mRNA, that is transported out of the nucleus for translation. So this step is called transcription, and the question is, what is the difference between what is that additional thing done in eukaryotic transcription? First step is splicing, second is capping, and third one is tailing. In splicing, the introns are removed and the exons are joined together. In second step, capping, methyl guanosine triphosphate is added to the fifth prime end. Okay, in tailing, it is added to the third prime end in the template. So if the fully processed hnRNA is called mRNA, and that is what transported out of the nucleus for the translation. Good. Okay, the next question. The flow of genetic information is shown below. Name the process. A is replication, B is transcription, C is translation, and D is reverse transcription. Okay, my next question. RNA is not an ideal molecule as genetic material because it is A and B. Okay, the OH group in the second carbon atom of the ribose is reactive and make it liable, and it is a catalytic and hence reactive. And for RNA mutation takes place very quickly. Yeah. The presence of lactose enhances the production of 3 galactosidase and other enzymes in bacteria. How will you explain this process? Okay, whenever galactosidase or the other two is given, you have to other two in the sense the permeases and transacetylases, you have to think about lac-operon. Lac-operon, the lactose is the inducer. The inducer binds with repressor protein, but repressor cannot bind to operator gene. The free operator gene induces the RNA polymerase to bind with promoter and initiate transcription. The 3 structural genes synthesize 3 mRNA to produce enzyme. Okay, so the lac-operon, the lactose is the inducer. And you have to know that when the lactose is added, it changes into glucose and galactose. Okay. DNA changes into mRNA. The repressor mRNA is changed into what? Repressor. If lactose is there, it acts as an inhibitor. Lactose, what he does? He acts as an inhibitor. if lactose acts as an inducer, inhibitor, repressor inhibitor, but it acts as a okay inducer. The inducer bind with a repressor protein, but repressor protein cannot bind to so that the repressor cannot bind to the operator gene, and the free operator gene induces the RNA polymerase to bind with promoter and initiate transcription. The three structural gene synthesizes 3 mRNA to produce enzyme. Okay, we'll skip that question. Okay, so stop codon is very important. Okay, this is the diagrammatic representation of central dogma. Observe the diagram carefully and redraw it, marking appropriate correction. The DNA to mRNA changes into DNA. The DNA is changed into mRNA. That is called transcription. And mRNA is changed into protein, which is called translation. It is there in the page number. Page number 98. In that it is given DNA, mRNA. DNA to mRNA, mRNA to protein. Then identify the diagram and explain. Identify the gram. It is the central dogma of molecular biology, and you have to explain that how it goes. In some cases, DNA is produced from RNA. Name the process and give example. Okay, that's an important thing. RNA to DNA is called reverse transcription. So what is mutation? Mutation is the phenomena in which results in alteration of DNA sequence and consequently result in change in the genotype and phenotype of an organism. So mutation is a phenomena which result in the alteration of the DNA sequence and consequently result in change in the genotype and phenotype of an organism. What are the different types of mutation? Physical mutation and chemical mutation. Very important question. Briefly write the method involved in the technique. Isolation of the DNA, digestion of the DNA by restriction endonucleases. Then separation of the DNA fragments by electrophoresis, the transferring of the separated DNA fragments into synthetic membrane, next hybridization using labeled VNTR probe, then the detection of hybridized DNA fragments. Where is the application? It's used in the forensic science. This is a very, very consequently asked question. A is transcription, B is translation. Explain transcription. DNA changes into mRNA. Information contained in DNA is copied down to mRNA is called transcription. Name of two regions, promoter and structural gene. Then this question. Observe the following diagram and answer the question. Diagrammatically represent the change take place when lactose is added to the media. That is what we have explained a few minutes back. But we have to diagrammatically show that. That's it. Thank you.